I talk quite loudly. Can you hear me?
Yes.
No microphone. All right. Okay. I think there's quite a good 2 lectures on regulation. I just changed my slides last minute because I had some slides on regulation, but I think you've heard enough. What I'm going to talk today to you over 45 minutes. It's just a little bit about the science around cell therapy, because I thought, I assume some of you are basic science researchers, so it'd be quite nice just to get what is happening out there. I will then give a situation of what we're doing in Singapore and what we're doing in the UK, for example, in trying to promote cell therapy as a country. And I know that this is under SPARC, and I was just trying to find out a little bit more, and I think the SPARC program is trying to stimulate cell therapy and trying to get it as an economically viable mode for the health system. On the other hand, you've also heard how expensive cell therapy is, and therefore I think cost has to be factored into any national health system. So I hope I can cover all of this within the next 40 minutes. I didn't bring my name card for some of you who might come. So my email address is there. More than welcome if you want to write to me, and it should be in your handout as well. Okay? I'm a clinical hematologist, so I will come from a quite clinical background. And as I said, I work between the UK and Singapore, and I run a cell therapy institute. So we'll start. I quite like to start always with a simple slide like this to show that actually more than 120 years ago, maybe, when they first started clinical blood transfusion, they connected a needle from one patient and put it from a donor to a patient, and you have fresh blood going through there. As Albert said, you know, if you work in the blood system, you naturally are really involved in cell therapy because you're dealing with cells. So actually, cell therapy is not new. It's been there for 120 and 30 years. And a lot of the rules, regulations, guidance, and understanding of advanced cell therapy at the moment has really been based on blood transfusion. I then move on to transplants, because transplants, I presume, are really the closest we have to an established model of cell therapy and was the framework upon which cell therapy came about. Okay? I'm quite involved in this organization, and I will talk to you a little bit later about this, but this is a conglomerate or an association of quite a lot of transplant societies, cell therapy societies, blood transfusion, and HLA typing. And you can see that, you know, we have now done 1 million transplants worldwide a couple of years ago. And this, of course, includes Taiwan. Taiwan has got a very thriving stem cell transplant and bone marrow transplant program. So bone marrow transplant is a form of cell therapy. It is well recognized, successful, and achieves cure. And because of that, and because of the experience of use, a lot of what happens within stem cell transplant is not regulated in the same way as advanced cell therapy, and rightly so. Now, this data just shows, and again, I've showed this slide before, I think for some of you in the audience, just to demonstrate that in certain diseases, using stem cell transplant, we are achieving 85 to 90% cure rate. So this is as good as it is. And we need to think about whether in the next 10 years, in the next 20 years, we can get advanced cell therapy to approach this level. I think for the last 20 years, we've been trying to address the issues of safety, how it is being produced, how to manufacture it, and these are important, including the cost issues and the regulatory issues. But we need to really answer this question now. Can we get the equivalent success rate? And those of you who know about CAR T cells, which I will talk about later, we are seeing that. So it's an exciting field that we're working in. And just a final slide on transplant, just to say that as medicine advances and changes, established modalities, including stem cell transplant, also changes. So I think that because we know how to do transplants better, the risk of doing a transplant now is much less than it was 20 years ago. We also have access to a lot more stem cell sources. We now not only look for brother or sister that's HLA matched, we can use cord blood, We can use haploidentical transplant, meaning from a father or a mother or a son or a daughter. When that happens, it immediately means that a huge section of the world population is now eligible for transplants. Because in the past, firstly, as you know, many countries in Asia have got small family sizes, so they can't do transplants if you've got no brothers and sisters. If you go to the unrelated registries, they are often dominated by a Caucasian or white population. So I think all of this has made transplants increase in number and across the world. I think that as we go on, we are going to do more and more transplants for non-malignant disease and inherited diseases. So things like thalassemia, things like sickle cell disease. And we're going to probably be doing less in cancers because at the same time, there's a lot of drug development and a lot of new novel molecules that are being used to treat cancers, which are achieving very good results. And it may mean that they are going to replace transplants. So in the same way that transplants are an evolving field, I think cell therapy needs to take the lessons on board from blood transfusion and transplant and move on. Okay? So those are the only slides I'm going to show on that, and then we'll move on now to cell therapy. And just one quick slide to just show that I think, this is quite an old review article, but it really demonstrates, I think, even at that time, that there are a lot of immune cells that we can work with that seem to be very important in controlling inflammation and in targeting tumors. And a lot of effort has been made in dendritic cells, as you've heard from Sam Shimotaka in the first lecture with Dendrion. And then a lot of people did a lot of work with natural killer cells. I won't talk too much about those two, but I will concentrate now on T cells, which seems to be the new big hope and seems to be the new thing that is coming along that will solve everybody's problem. And I will touch on that. Let's skip these two, just to say how it works. So CAR T cells, I think some of you I assume some of you might work in it, some of you may not. So I'm going to just quickly do a small introduction and talk about how this has really changed the face of cell therapy. Now, so this really shows a T cell or an engineered T cell engaging the tumor. It has been so important that the ASCO, the American Society of Clinical Oncology, when they reviewed the top 5 advances in cancer treatment, you can see that it spans over the last 50 years, and this includes CAR T cells. Why? Because we are seeing success rates that we have never ever seen before, and in some ways, for the first time, we're seeing cell therapy achieve better results than traditional chemotherapy or even novel drug treatment. And I think that's very, very important. This was the first paper published by the University of Penn group, Carl June, and it really showed that in a patient with chronic lymphocytic leukemia, so these are lymphoid cancers, with just the use of these CAR T cells in patients who were refractory or not responding to any other treatment available, we saw complete remission. So it wasn't even some response, it wasn't even partial response, it was complete remission. And these were patients who really had no other hope left. And this has been reproduced in multiple trials across acute lymphoblastic leukemias as well as non-Hodgkin lymphoma, and has led to the FDA approval of 2 drugs to treat these lymphoid cancers, but they are costing $500,000 US for one administration. When you've got $500,000 to treat one patient, every country needs to think how they're going to afford it, because no country can do this for everybody. This slide just demonstrates 2 things. This is just an introduction, so I can't go into too much detail, but the bottom slide just shows what a CAR T cell is. Where's the pointer? Okay. So this just shows a CAR T cell. We know that a T cell, okay, is very powerful, but a T cell to engage in an immune response a T cell needs two things. It needs a specific antigen, and it needs to recognize it within the HLA context that it is associated with. So this makes it much more restricted, but it's very powerful. On the other hand, B cells or antibodies are different. Antibodies recognize a common antigen, CD20, CD19, CD3. So if you make an antibody, it targets quite a big range and is independent of HLA type. If you're able to combine the two using a CD19 that recognizes on the backbone of a T cell and how it signals, you end up with a very powerful molecule. And on top of that, then the newer generation of these molecules have incorporated other co-stimulatory domains. If you go on to the first slide, this is what is involved, okay? This is an autologous therapy. You've heard from the last two lectures the autologous therapy and some of the ways that they bypass regulation. This is a complex autologous therapy, should never be classified or in any health system possible, should never be considered as safe and not regulated just because it is autologous. Why? Because it is genetically modified and it has got problems. But you think about it, to make this cell, this is the process that you need. You need to be close to the patient, you need to collect the cells from the patient, and then you need to bring it to the laboratory to activate it, to transfect or transduce the virus into this, and then you need to expand it, and then you need to harvest, you can freeze it down, and then give it back to the patient. It's time consuming, and to some extent you can see that it needs to be quite close to the patient. Okay? You can't just do it 2,000 miles away and ship it because logistically it's impossible. So again, this points that if this therapy becomes commonplace, how do countries or how do hospitals adjust to this new way of thinking? And this is the same slide again, just showing the steps involved, going from apheresis, T cell selection. So some of it we already know how to do. Genetic transfer, you need to expand it, cryopreserve, and give it back to the patient. When this was first done, it was done in kind of open systems in cell culture flasks. As time has moved on, and I think industry has a very important role to play, and that's why the SPARK program, I guess, is important. You can see industry keeping up and trying to provide solutions, many of it automated, to try to reduce variability, the time consumption, the manpower, and hopefully some of the cost involved. So the promise of cell therapy is as you make this more and more widespread and more people use it, that technology comes in to reduce the cost. We have seen this when we have been sequencing the whole genome. If you remember 10, 15 years ago when they sequenced the whole genome, it cost quite a lot of money. These days it takes 20 minutes. You can sequence the whole genome at a fraction of that cost. Will cell therapy go that way? I don't know. But that's the hope. Okay. Now, the next slide is quite busy. I just want to highlight a few points. The few points are that there have been various trials, all of them most of them pharmaceutical supported, although originally it originated within the universities itself. So this again, I think, is something that the SPARK can take note of because it is a homegrown technology that came from the University of Penn, brought over by the pharmaceutical companies, and lo and behold has been a changer in medicine. These are across a couple of trials. You can see that unlike drug trials, not many patients are treated. You know, drug trials sometimes, I mean, if you look at some of the cardiac trials, they run into thousands of patients to get approval. Here, the total experience has been less than 150 at this time. Okay? But look at the complete remission rates. They are in excess of 80% in most of the trials. Okay? And that is remarkable in a subset of patients that are, again, really at the end of your medical therapeutic line. If you look at the success rates, look again carefully at the reported deaths. There are people dying from cerebral edema and dying from something called CRS, which is cytokine release syndrome. So with efficacy comes problems, and I think that's why regulation is important. So we learned that these cells, although they're very, very powerful, induce, because you know anything, when you activate your immune system, you're going to get a flood or a torrent of cytokines coming out. And those cytokines can be detrimental to the body. It causes pulmonary edema, hypertension, tachycardia, vasodilation, fever, and patients die. And therefore, it also tells you how important it is for the clinicians who are working in a novel field like this to be able to quickly learn novel side effects and to tackle it. Because if they did not do this and patients continue to die, the whole trial would have been stopped. The regulators would have stopped the trial, and we would not have seen CAR T cells approved. But the UPenn people who ran the initial trials were quite astute, and they realized that a lot of these they measured the cytokine levels, and they found that IL-6 was hugely high. And so they introduced anti-IL-6 or tocilizumab, and they found that this stopped the CRS and patients survived. Understand? So it was very important. So you can see drug development, therapy development, wonderful success, and then you always get some side effect and how you deal with those side effects. We're still trying to come in terms of how the neurological toxicity is, because we don't fully understand it. The problem, though, is that it appears that for it to work, you do need to get some degree of this cytokine release. And that's logical. If you don't get cytokine release, the cells are not doing anything. So you have a modality that's equivalently powerful, but equivalently perhaps a little, well, not a little, but toxic as well. Okay? And that's why it's called a controversial cure, because patients do die. And this is to do with the cost of manufacture. If you look at the world now, this is the state of the CAR T cell that is going on, so really across the world, and you can see that some in Japan, China is enthusiastically kind of taken this on board. We are just gearing up in Singapore to do our first CAR T cell trial in about 2 months' time. Australia, and then that's the European Union. So I think wide acceptance across all countries, and I think will continue to do so. So it's worked fantastically in blood cancers. Blood cancers are a small part of cancer. You've got lung cancer, stomach cancer, breast cancer, much bigger population. How does it look? There are CAR T cell trials looking at currently being enrolled, but you just look at the amount of green in each. The top figure is for blood cancers. The green basically shows success rates or complete remission and partial response. You can see that in blood cancers, you've got quite fantastic responses in the lymphoid malignancies, less so in your myeloid, and then we're beginning to understand and do them in myelomas as well. But look at the comparable situation in solid tumors, a lot less green. Okay? The only exception is an antigen called GD2, which is found in neuroblastoma. So the neuroblastoma story looks quite good, but look at the number of patients treated, only 90. So I think we still do not know, in other words. And I'll just quickly run through in one slide why the problem is in solid tumors. The problem in solid tumors, unlike blood cancers, in blood cancers, your cancer cells are swimming in the blood and bone marrow. They're there. In a solid tumor, they're embedded in an organ, or the tumor grows into the organ. Number two, blood cancers, as you know, lymphocytes, myelocytes, they express very, very reproducible antigens, whether it's CD19, CD20, CD3, CD33. That's how we do flow cytometry. But in solid tumors, it's a little bit more difficult to identify the antigen that is needed. Number two, you need to get these CAR T cells into the tumor. When you're injecting it into the blood cancer, they're already in there, seeing the tumor. But for solid tumors, it's a slightly different problem. We know that solid tumors have got an environment that's very, very suppressive and does not allow the immune system to work. So you need to bypass that as well. You need to somehow activate these CAR T cells. You need to make them proliferate when they go in there to combat the tumor microenvironment. And then finally, if these solid organ tumors, if these tumors happen in vital organs like the lung or the liver, you can't have too much collateral damage because you can't destroy the organ, right? Okay, because that's a very important thing. So I think trying to use CAR T cell in solid tumors is probably a lot more difficult, but there's a lot of research going along with it. I think this is good. Now, so I think this, I brought in CAR T cells, and I'm sure that the other lectures, maybe along the way, we'll talk a little bit more about this, but has really changed the face of cell therapy because clinicians or researchers who had previously not believed in cell therapy for many, many years are now understanding that this perhaps is the future of medicine, and that perhaps we are already seeing what I would say a cell-based medical therapeutic system. We are no longer looking at our standard chemotherapy drugs and standard drugs. We're looking at novel drugs like checkpoint inhibitors, targeted therapies, and we are looking at cell-based medicine. Okay? And CAR T cells are not the only way to go. T cells are also used in other ways. And I'll just show you two examples. Okay? Remember I said that in solid tumors, CAR T cells have a problem going in for various reasons. The other way of attacking some of these solid tumors is to use T cells that target viruses. Because we know that a viral specific T cell response is perhaps one of the most powerful immune responses in the body, and it's also reproducible, and it's something the body is used to because we have evolved that way. So if you've got cancers that are driven by viruses, and these are EBV-driven lymphomas, and in Asian countries, nasopharyngeal carcinoma, NPC. If these are driven by viruses, you automatically have a viral antigen you can target. And if you develop T cell therapies against it, it can be very powerful. And I'll just show you two examples. So the first example was just published 3 weeks ago in Nature Medicine, and it just tells you how complex cell therapy has become. This is from the NCI in America by Steve Rosenberg, who really, I think, is probably the pioneer of all T cell therapy. He looked at patients with breast cancer. Again, these are patients who have failed all treatment, including hormonal treatment and so on, end stage. What he did was to take the tumor and bash it up, basically, looked at the DNA and RNA, and did whole exome sequencing and transcriptome analysis. So really large data analysis, okay, which really all big labs these days perhaps do need to analyze big data to try to get the mutation and what these antigens are. So really you're searching for these antigens. Having searched for these epitopes and antigens, he then cloned it into mini genes and transduced this into the same patient's dendritic cells. So all of that is genetic, computational biology, and informational technology. At the same time, he's isolated lymphocytes, tumor lymphocytes from within the breast cancer, the cell therapy world, exposed them to these dendritic cells to see so that the ones who recognize these antigens expand out, and then he has then grown up these T cells in huge quantities. If you look at it, in the CAR T cell world, you were giving million per kilogram, a million. Here you're giving up to probably billions, okay, near billions, so 1,000-fold more, and giving back to the patient, adding on checkpoint inhibitors, anti-PD-1. This method was successful. These patients, or this patient, went into complete remission. So again, another success story, and importantly, what does it show? It shows that it works in solid tumors. It shows that it works even in breast cancer, which is always thought, a common cancer needs to be targeted. But what do we need to get there? We need a combination of cell therapy, which is expensive, computational biology and IT, which is expensive, plus approved drugs out there which are not cheap. So how do we manage these costs is a problem. So that's one way of looking at it. The other way, just very, very quickly before I move on to the kind of more policy part of my lecture, is to, so this is the current trial we're running in Singapore. So just to say that, nasopharyngeal carcinoma, I've told you, EBV driven, we can find the virus. So we now produce these dendritic cells using a STEAP137 ligand because we think that these produce a DC that is more immune inflammatory rather than the standard GM-CSF and IL-4. We then generate T cells from this and look at the response to NPC. The trial is ongoing and hope will be successful. But just to demonstrate that CAR T cells are not the one and be all, I think other strategies of T cells are important. And yesterday I was just listening to a lecture on NK cells, and that looks promising as well. So I think that all of you working out there in immune biology, carry on, and something like this may happen. I'm just going to very quickly deal with, I've dealt a lot with the autologous pathway, using something from patient, giving it back to patient, and it all looks expensive. The other way is an allogeneic product, and an allogeneic product means you can produce in large batches, store it, bank it, and give it back. It seems easier. And one of the ways of doing, one of the poster boys, or one of the shining examples is MSCs. And you can, nowadays people use MSCs for everything, for graft versus host disease, for cardiac regeneration, neuronal degeneration, and you can get MSCs, as you saw from Albert's slide, from liposuction. You can get it from bone marrow. It is easy to multiply. You don't need to match them. They can cross HLA barriers. You can freeze them. You can expand them. Easy. And actually, it's not expensive to produce. If MSCs were the answer to this world, it would be great, because it's actually not too expensive. It can be treated like a drug. The problem is that we still do not know how to fully define what an MSC is, I think. We still do not fully understand the mechanisms of action. And so I think if we do not get our science right, it's very difficult to imagine how we're going to carry on in this field. But this is nonetheless promising. And how do we grow this? And I'll just leave it on to the next thing, which I thought I wanted to show because Thierry's in the audience and Taiwan was involved in this, so I thought it would be nice to show this. So we've been looking, Thierry and I have been looking at this for a while now, trying to look at how platelets and the products from platelets, how they can be used to support cell therapy growth, because they are very rich in nutrients for cell therapy immune cells. But not only that, as you've heard, I think that in Albert's lecture as well, that people are giving back platelets for joint injury, for tendon injury, for muscular injury. So it's also used in medical practice as well. So the question is that cell therapy not only looks at the immune cells, but also looks at other things, including how you can manufacture these cells using growth factors, and some of them involve platelets. So we recently had quite a successful meeting, and I think Taiwan is represented there. And what we basically were trying to do was to understand the basic science of platelets, how they regenerate, and from there, again, understanding the science, making sure it's safe, how to qualify and standardize this, and how to use this. This can be applied to any cell therapy product. This can be applied to a blood-derived product. Even looking at plasma, red cells, you can even think about this to some extent. Okay? So this is not intrinsic to this, but I think you can use this as a framework. And then finally, of course, and I won't dwell into this because I know there's an IPS lecture from Japan, so I really should not talk about this. But I've dealt with autologous immune products against cancer. I've dealt with an allogeneic product, which is more wide-ranging, not only in cancers, but across regenerative medicine. And the last thing is stem cell-based medicine. How do stem cells work? And what can they do? And the potential is great, but the problems, the potential problems may also be great. I won't go much into iPS cells, but as you know, in Japan there is a trial ongoing for macular degeneration Why macular degeneration? More and more common, especially in developed countries. No drug therapy available, progressively leads to blindness. So something that really needs treatment. And so lo and behold, iPS cells have been used to try to regenerate the retinal stem cell to reverse this. First patient treated went well and seems to have achieved good results. However, the second patient, when they did the iPS and looked at it before giving it back to the patient, they found genomic mutation within it. We don't know whether this would have done anything clinically in the patient. But I think the Japanese, rightly so, felt that the instability of the genome made it a little bit difficult to say that we can inject this into the patient, so they did not, and the trial was stopped for a while, but it's restarted again, and I think it's ongoing. So again, it demonstrates to you how novel technologies can evolve using stem cells, but I think you need to put safety always first before you deal with efficacy. And don't forget that we arrived at this CAR T-cell success rate after 20 years of trying to deal with safety. I won't deal with this. Sam has dealt with this comprehensively. So, the last, how long more have I got? 10 more minutes. Just stop. Okay, the last 10 minutes, I will try to deal a little bit more with policy and how we integrate this. So, we have, on the one hand, I've shown to you expensive autologous products, but perhaps with excellent clinical outcomes. On the other hand, there are also products that can be easy to produce, like mesenchymal stromal cells, can be treated almost like a drug, a wide spectrum of use, but at the moment, although it appears to be safe, we don't have the same amount of clinical efficacy data. And then we've got stem cells ranging from embryonic to iPS, and within that, I think you have to answer the ethical issues of certainly of embryonic stem cells. You need to answer the scientific questions and also the potential genomic and stem cell differentiation capabilities and whether you can form tumors and the like in stem cells. And when you're dealing this for countries, you need to think about cost, you need to think about how effective they are, and you need to also think whether there are alternative ways of treating the same disease. And this really should stretch right across from academic research to the translational stage to what the country, the university, and the hospital can provide as infrastructure for you to develop these therapies. I would say that really, in my years in cell therapy, over the last 5 years, I see more and more countries develop a more national strategy towards cell-based medicine. And I think the understanding, especially, I can tell you that although cancer therapy, cancer cell therapy is perhaps more advanced at the moment, I would say that the larger market and probably what will change the face of medicine is regenerative medicine. Because as countries develop, we all have aging populations. With aging populations come chronic diseases with chronic care, no effective drug therapy, and it's expensive to look after these patients. If you think about how expensive it is to look after a stroke patient with a neurological disability, you need to factor this against what you can potentially improve in their function and perhaps not needing the supportive care that they require. And so countries are now looking at how they're all looking at research institutions and universities to come up with the science and the homegrown product so that you don't need to buy some of these expensive products, how to commercialize this to achieve economic potential, thinking about cost, but also making sure that everybody in the population has access to this, so equity of care. And how do the regulators kind of follow this and work with the national health system? Because you are right, I think all of us agree at the moment, it is still expensive these days, it is not cost efficient. And I'll give you a few examples. In the USA, for example, the USA have started this network called PACT, which is called Production Assistance of Cell Therapy. So these are certain universities with large GMP labs, And what they've done is that the whole country, you're going to produce cell therapy products, you can feed it into this PAC system. They provided funding so that you can make these cells for, not for free, but then you can therefore make these cells with governmental funding. In Canada, they've got something called CCRM, which is a Center for Commercialization of Cell Therapy, and this is based in Toronto, and this is trying to engage industry with universities to build up homegrown cell therapy markets. I would think a little bit like what Spark are trying to do, and the same thing as what Singapore is trying to do as well. And the same thing happens in the UK. It's called Catapult. Okay, so Catapult works. So they come to us, for example, universities, and say, okay, you've got a fantastic research going on. We would like to help you to translate this into a clinical trial. They will then look for a manufacturer for you to manufacture these cells, and they might even partner you with a commercial company who may want to take on that product. They will also approach pharmaceutical companies and say, look here, you have got a nice product that you want to do it. Let me work with the clinicians to see how we can facilitate the trial. And the purpose of this is to try to build a healthy ecosystem for cell therapy. And within California, as you know where Hollywood is and everything is, regenerative medicine and trying to look younger and stop aging is extremely important. So CIRM has had a huge amount of funding, and a lot of it from private investors, because the billionaires these days want to live forever. So a lot of anti-aging therapies are all going on in the California Institute of Regenerative. I think I've talked about this. So I've talked about the UK. So I think the UK, the only thing to sort of say is that the one thing about the UK is that it's a national health system. Nobody has to pay for their health care. If nobody has to pay for their health care, a product that comes in that costs $500,000 is going to be a problem. Okay? So they're trying at the moment to work out how to get CAR T cells available for the UK. And so on the one hand, it's difficult because it's a free system. On the other hand, it's been useful because they can negotiate with the companies directly as a country. Rather than as a hospital or as a regional group, they can go to the country and say, look here, perhaps for the UK, you need to do this at this price, because otherwise we can't do this, and in what kind of groups of population. Okay. In Singapore, just to give you a few things, the Ministry of Health have put up quite a nice program to support cell therapy. So at the moment, I run a smallish facility where we've got about 8 clean rooms. We're going to expand this into 22 clean rooms in 2022, and this will try to engage some of the pharmaceutical sector as well, to try to create an environment for hospitals that can engage with us and pharmaceuticals engage with them. At the same time, the national funding and grant awards have recently understood and have included the translational cost of cell therapy, meaning how to translate your research into a GMP-grade material. The Ministry of Trade and Industry is collaborating with academics and biotechs to make this an economically attractive option for Singapore. And we're trying, and then finally, I think two things that we forget is that we need to train up regulators, or not that we, but regulators need to be trained up to deal with this modality. I'm sure Albert has a lot to say. And the second thing is how to train up laboratory staff and clinical staff to deal with the complications of cell therapy, but also how to manufacture these things. And the idea in Singapore at the moment is to try to build up a training center so that anybody in the region can come to us for training. This is where the cell therapy facility is housed. It's also where the National Blood Bank is. It's also where the equivalent of the FDA is. So it's quite nice, everything housed in one single unit. That's the lab. We recently acquired the Prodigy system from Kenny as well. And then just a few slides showing, as you know, Singapore has invested, 2 more minutes. As you know, Singapore has invested heavily in the biopharmaceutical sector. It contributes 4% of Singapore's GDP at the moment, and lots of biologics and drug products employing lots of people. So I think because of that, they have realized that cell therapy could perhaps come along this route, and I think that's why we have been quite lucky, I think, working with this team. Last 2 slides. First of all, just to reiterate what the problems are for autologous versus allogeneic. In autologous, it's like a personalized medicine. You take your cells, you make it something different, and you give it back. Okay? And anything that is done like this is always expensive because it's personalized, it's bespoke. Allogeneic is you can manufacture it from one. It's a little bit like a blood product. You know, you can, well, blood product is more. It's more like a drug. You can just get it from a few donors, make a lot of cells from it, and you can bank it. Okay? And so the manufacturing for this, okay, for autologous, to some extent has to be quite close to the hospital because you need to be close to the patient. Whereas in the allogeneic world, it can be quite centralized. You can make it in America and ship it across. There's no problem. And I think you referred to this because I'm running out of time. And just the next slide. So how do we convert an autologous process that is expensive? At the moment, we're still doing this in cell culture flask, lots of people working on it. And the hope is that, and this is already coming aboard, so I think the industry is understanding this. So I think the engineering and the technology industry is very important, and they're coming with solutions to remotely monitor that can feed in cytokines, that can feed in change media, all robotically and automatically. At the same time, in the allogeneic world, at the moment, we're using big flasks. We're now thinking about big bioreactors and microcarriers to do this. And so, what you need to do is that you can scale things up, increase efficiency, and reduce, then you can kind of try to reduce the cost of the drug sold while not compromising on safety, and you need to continue to demonstrate robustness of product. Does that sound familiar to the blood transfusion world? And so, this really is my last slide. For a successful cell therapy ecosystem, you really need the clinical centers to validate the clinical bits of it. You need the regulatory leadership, but you also need the enabling and innovation associated with it, and finally resulting in these therapeutic assets for universities, for companies, for countries involved. So thank you very much.
Thank you for a very nice talk. So now we have a lot of understanding of a lot of manufacturing aspect and healthcare and what we need to do. Short question, anyone?
Yes.
Hi, good morning, Dr. Koh. I'm Mabel. I'm from Cancer Therapeutics, which happens to be a Singapore biotech company. I think your lecture was very inspiring. I think you mentioned a lot about national and policies perspective. So I just would like to get a comment from you on how biotech companies like us, as well as the hospital institutions, how do we prepare ourselves, and how do we move towards, overcoming a lot of these operational challenge and, changing the mindset to embrace this kind of future therapies.
So for those of you in the audience, Tessa Therapeutics is a homegrown Singapore cell therapy company. Seems to be doing very well. They are using a viral-based, correct me if I'm wrong, okay? They're using a viral-based strategy to treat a viral-based cancer. So again, things like EBV-related cancers. And they're running a big T-cell trial. They always call it the largest phase 3, international phase 3 trial in the world. And it is an autologous way of manufacturing. In response to you, I think the government, the Ministry of Health has already recognized that we need companies like you to foster this ecosystem. So this new cell therapy facility that's being built, there are already discussions with Tessa Therapeutics as to how they can interact with the academic environment. Okay. Already, I think you have got clinicians that are running some of these trials within Singapore, so the interaction is there already. So I think that, and then don't forget the researchers are now accessing grant funding that will also have inbuilt into it the grant for translational trial as well. My lab has got seed funding now to support some of these homegrown technologies. So all of this can feed into Tessa. Tessa can then also leverage on it. Any other, please? Yeah, please. Yeah.
in Singapore. Yeah, it's very similar like Taiwan Spark. I'm very interested about how the government support the CCRM to support the industry translation from academic science.
Yeah,
Yes, thank
yeah.
you.
But having said that, I mean, I visited the CCRM in Ontario, Toronto recently, and it's always a nice idea. So when CCRM came about, and even when Catapult in the UK came about, they had a big funding from the government. The Catapult had, I think, £50 million funding for 5 years. CCRM had big funding. The problem is how to demonstrate those results after 5 years, and how do you get your next level of funding? Sorry, Jane is looking at me nodding because that is your main problem. Because 5 years is not a long time because you need 5 years to set up the structure to engage people, and then the time is up. How do you get your next tranche of funding? See, the Canadians were they were in danger of actually dissolving. They were rescued in the last minute by GE Healthcare. So they had a big pharmaceutical company come in and kind of help them. And Catapult had big issues as well, and so they're looking at a slightly different module system. The problem with Singapore and Taiwan when we're dealing with this is that we do not at the moment have the critical mass of local companies and big enough companies that can support this. You know, if you're big companies, they automatically go to the US, the EU. I think Japan obviously has a good enough infrastructure, and because of the IPS technology, I think they can manage it. But we don't. I think those are the bits that we struggle with. Isn't that right?
That's always an issue. Singapore actually has a Spark. Singapore,
yes.
Yes,
Yeah.
And actually Australia too, Japan. Kyoto, I believe, is the first Spark. Tokyo University only has the educational part, doesn't have the commercialization education part. So I think every one of us are on the same boat. We are trying to fight through demonstrating that, you know, it is possible. Give us time, give us money, and then we can prove to you we can be helpful to the patient. Thank Dr. Goh for the nice discussion.